Hello there, welcome to Gaurav Tutorials. In this video, we will be discussing about phases of growth. But first, let us have a brief recall of what we studied in the last video. In the last class, we discussed about the growth being measurable. This measurement can be done with respect to the surface area. It can be done with respect to the length of the plant. It could be done with respect to the weight, volume, number of cells, and with respect to the cell size. So in today's class, we will be discussing about phases of growth, and this growth in the case of the plant takes place in three phases. First of them is the meristematic phase, followed by the elongation phase, that is the second phase, and lastly there is a third phase, that is maturation. So let us dive into all of these three phases one by one. The first phase is called as the meristematic phase. It is also known as formative phase. The first phase of the growth in case of the plant, that is meristematic phase, is also referred to as a formatic phase. So what happens during this phase is that the cells of the meristem will divide and they will add new cells in the structure of the plant along the length of the plant, in fact. So these parent cell of the meristematic and these newly formed cells are different, right? They have different structures. So first let us talk about the cells of these meristems. Now when you generally talk about the meristems, these cells are usually very thin walled. They have a very thin wall as they have to undergo a constant division. Okay, they are thin walled and they have a very dense protoplasm. Now this dense protoplasm helps them to actually cause a division to the daughter cells and providing enough cytoplasm for both the daughter cells. Excuse me. There is a dense protoplasm followed by a smaller sized vacuole. Okay. As these cells are only having the function of division and there is no necessity of any stored food for them, the vacuole in them seems to be very small. The size of these cells are isodiametric, which means roughly when you measure these cells along any direction, you'll find the cells having almost same diameter along its area. Now they have because they have to cause division, they have a very large nucleus. The nucleus seems to be very large, and yeah, these are some of the few features of the meristematic cells. Now what do these newly formed cells do? These newly formed cells enter into the phase of elongation. So this elongation phase follows the meristematic phase. Right. Now what do we actually find is there is the cell of these region, that is elongation region, or the cells which are undergoing elongation, are having rich amount of water, rich amount of solute. They are very rich in solutes in them, right? The minerals and the ions and the biomolecules are very much in higher concentration with respect to the meristematic cells in case of these elongating cells. Now as they are rich in solute, what will they do is they will absorb more amount of water. They'll absorb more water, and while the water is entering into the cell, the water also takes up other solutes along with it. Okay, so if this is my cell, okay, this is my cell, and what do I find in the cell is that these cells are having lots and lots of solutes in them, okay, inside the cell. Whereas outside you find the concentration of solute being lesser. However, the amount of water in the outer region is far more greater than the amount of water present inside the cell. So what does it do is it establishes a gradient. There is a difference of the water created between this outer and the inner region of the cell. This allows the water to enter into the cell. Okay, and when the water enters the cell, along with it also takes some of the solutes which are there in the outside, right? So this constant input of water, okay, constant inflow of water, let me put inflow here. The constant inflow of water and the solutes into the cell, what does it do is it ensures that the turgor pressure of the cell increases. Okay, the turgor pressure of the cell rises due to the entry of the water and the solute into the cell. Now, as a result of this, the shape of the cell remains intact. Okay, the shape of the cell remains intact, and due to increased turgor pressure, excuse me, due to increased turgor pressure of the cell, we find there is a development of the cell wall. Okay, the cell wall develops here. Okay, the cell wall which is formed here is of basically primary nature, and here the cell wall which was formed here in the meristematic region had primary cell wall. It was a primary cell wall. During the elongation phase, this cell wall turns secondary. That means more amount of cellulose gets deposited here. Okay, so there are two theories proposed for this deposition of the cell wall, cellulose in the cell wall. One of them is intussusception. Intussusception. Now, what does it indicate? What does it mean is, if this is my cell wall which had some amount of cellulose in it previously, will get cellulose deposited within the, like, along this cell wall, right? It will be developed between the cell wall. The new cellulose will be developed between the particles of the old cell wall. Particles of old cell wall. Okay, the other theory about this deposition of the cell wall is referred to as apposition. It is called as apposition. In the apposition, what will happen, if that is my cell, and this happens to be the cell wall of the plant, what will I find? That the new cellulose particles which are to be deposited will be deposited on the inner side of the cell wall. Okay, the cell wall will find deposition of new cellulose along the inner side. Okay, the particles of the cellulose will be deposited on the inner surface, right? So there are two theories proposed for the deposition of the cellulose in the cell wall. First is intussusception, which states that the particles of the cellulose are deposited between the particles of the old cell wall, and then there's another theory by the name of apposition, which states that the particles of the cellulose are deposited on the inner surface of the cell wall. Right now, as the cell is growing, it is storing a lot of solutes, a lot of biomolecules in it. We also find that such cell which are undergoing elongation develops a large vacuole. Okay, they develop a large vacuole, and we find that the protoplasm in such cells occupies peripheral region. The protoplasm occupies peripheral region. Now I would take you to this diagram of the cell. So what will happen if this is my nucleus? I'll find there'll be a deposition of the large vacuole in a plant cell, and as the vacuole enlarges, what does it do? It pushes the protoplasm into the sides, that is the peripheral part of the cell, right? So that's your elongation part or elongation phase. Coming to the maturation phase, which happens to be the last phase of the growth, we find that elongated cells which have entered the maturation phase will start to take up fixed form and fixed function. They take up fixed forms and function. So this means by which the cell takes up a fixed form and a fixed function is also referred to as differentiation, right? Differentiation, the process by which a cell takes up a fixed form and a fixed function, is referred to as differentiation. And generally there are two types of differentiation that are to be taken into consideration. First of them is a structural differentiation. So when we talk about structural differentiation, what does it mean is the shape of the cell, the size of the cell, and the internal constitution of the cell will become fixed, right? Once the shape, the cell of this, the cell that has entered the maturation phase undergoes structural differentiation, I would find there would be changes in its shape, in its size, and its internal constitution. However, if the cell undergoes other type of differentiation, that is physiological differentiation, in that case the cell will acquire fixed function. It will occupy fixed function. So when the cell takes up a fixed form, I will call such a differentiation as the structural differentiation, and when the cell takes up a fixed function, I will call such a differentiation as a physiological differentiation. Now once the cells have taken up fixed shape and fixed function, they become specialized. They become specialized and become permanent cell. They take up a fixed shape and a fixed function to become a permanent cell. Now I would like to mention few of the examples of these permanent cells. Some of them are the fibers, some of them are tracheids, the other becomes vessels. And if you could have a brief recall of these fibers, tracheids and vessels, you would be able to understand that these are the parts of xylem. Similarly, they could take up the functions. They could become a permanent cell into a form of sieve cells or sieve tubes. They can turn into parenchyma. They can turn into something called as collenchyma. Similarly, they can also turn into sclerenchyma. I mean, what not? They could turn specialized, and they could take up a fixed shape and function to become a permanent cell. Some of the examples which are listed here, right? So I would actually want you to understand that during this phase of maturation, a cell has attained maximum size in respect to its cell wall thickening, and also the protoplasmic modifications that were to be undertaken by the cells are completed during the phase of maturation. No further changes would take place once the cell has become matured, right? So this point of differentiation, or this length of time, the time interval from this meristematic phase to the maturation phase, okay, the length of time that a cell stays in the maturation phase from the meristematic phase, I mean the distance or the length of time that a cell spends from this meristematic phase to the maturation phase, is referred to as grand period of growth. It's referred to as grand period of growth, right? So I'll just take a pause here and just undergo through, like, we'll just go through a brief recap of what we studied in this video. Is there are different phases of growth, starting from the meristematic, proceeding to the elongation, and lastly the maturation phase. We also talked about the fact the meristematic phase is also referred to as a formative phase, during which the newly formed cells are the new cells are formed from the meristematic cells. We discussed about the features of the meristematic cells. Then we pushed these newly formed cells from the meristematic cell, which were formed by the meristems, into the elongation phase, whereby they developed lot of morphological differences in them, and got the solutes and the water from the external environment into them. We also subjected these elongation cells, the cells of the elongation phase, to the maturation phase, whereby they took up the fixed form and shape and function. The fixed form was referred to as structural differentiation. The taking up of fixed function was referred to as physiological function, right? So yeah, lastly, the length of the time that a cell spends from the meristematic phase to the maturation phase was called as grand period of growth. So that is it, guys, in this video. I hope you would have understood what was being conveyed by the video, and if that's the case, you can hit the like button. You can also share the video with your friends, and if at all you find this channel to be useful for your further studies, you can also subscribe to the channel. In the next video, we will be talking about the growth rates. Till then, take a very good care of yourself, and yeah, God bless us all.